Good morning, everyone. Thank you for the lovely presentation. Today I'm going to present the work entitled Sex Estimation in an Adult Skeletal Remains, a Geometric Morphometric Approach using the Auricular Surface. So, the estimation of biological sex involves the classification of an individual as male or female based on their skeletal characteristics. This step is essential for elaborating the biological profile of human skeletal remains from archaeological contexts and studying human population dynamics and cultural behavior. For example, this parameter enables the assessment of sex-related differences in mortality rates and provides insights into sex-specific mortuary practices in past societies. It has also been valuable in studies regarding migrations, socioeconomic transitions, or the evolution of sexual dimorphism in the human species. So, sex estimation currently relies on three main approaches. Morphological, which involves the visual assessment of the shape and size of specific anatomical features, typically from the cranium and pelvis. Morphometric, which compares measurements and indices of certain anatomical elements and analyzes them quantitatively. Geometric morphometrics is included in this category. And finally, biomolecular, which includes DNA analysis and proteomics. These approaches are generally not applied to immature individuals except for biomolecular analysis, as sex estimation is considered unreliable before puberty. Several studies suggest hormonal differences between males and females may contribute to early sexual dimorphism, leading researchers to explore ways to distinguish between non-adult males and females, particularly through the ileum. Non-adult sex estimation through the auricular surface can be divided into three categories. The first focuses on visually assessing the auricular surface anatomical features. The second involves combining conventional morphometrics with morphology. And finally, the third category applies two-dimensional geometric morphometrics to the partial outline of the auricular surface. So, bearing this in mind, and what information the estimation of biological sex in adults can bring for the study of past populations, the present work addressed the following questions. First, can three-dimensional geometrics be used to detect sexual dimorphism in the auricular surface of infant skeletons under the age of 7? And second, is it possible to apply this methodology to archaeological collections? So, to address our questions, we developed a 3D geometric morphometrics methodology to estimate the biological sex of infants based on the auricular surface. This technique was tested and validated with 46 cilia from the Lisbon Identified Skeletal Collection, and then applied to 8 Mesolithic infants of unknown sex from the Mush Skeletal Collection. Our study focused on infants aged 0 to 6 years, excluding those with significant postmortem damage in the auricular surface. Samples were categorized into 3-year age groups, with those under 1 year placed in a separate category. To verify the age at death of the reference sample, the method proposed by Molson and Cox for the maximum ileum length was used. For the archaeological sample, we relied on centroid size from the reference sample due to fragmentary specimens. Following the GM methodology, the first step involved 3D bone digitization. Each specimen was also photographed and the maximum ileum length was recorded. Afterwards, 4 landmarks were manually placed at homologous points across all specimens, and 22 semi-landmarks were positioned along the auricular surface outline. The R-package geomorph was then used for standard geometric morphometrics and subsequent statistical analysis. So, analysis of the reference sample showed differences in the size of the auricular surface in 2 age categories. For individuals aged between 1 and 3 years, females had a larger auricular surface compared to males. In contrast, for the 4 to 6 year age group, males exhibited a larger auricular surface than females. Despite these observed differences, the Mann-Whitney test did not show that these variations are statistically significant. Regarding shape, independent analyses were conducted for each age group, revealing differences between males and females only in individuals under 1 year of age. The Permanova performed on the PC scores that explain around 95% of the total variance showed no statistically significant differences. However, visualization of PC1 and PC2 revealed sex-based differences in PC2, despite not being statistically significant. The biological sex of the archaeological sample was estimated based on specific analysis for each age category. Among the 5 archaeological individuals with an age at death of less than 1 year, only 1 is included in the female group. The other 4 individuals did not fit within the ranges of the reference groups. Based on the analysis, it was suggested that the 3 individuals closest to the minimum values of PC2 could be male, while the individual closest to the maximum PC2 values might be female. So, the analysis of the auricular surface size from the Lisbon sample showed a gradual age-related increase with different growth rates between sexes. Differences in auricular surface shape were evident in males and females under 1 year, suggesting early sexual dimorphism. These findings may be related to mini-puberty, which occurs during the first life due to the heightened production of sex hormones. In older individuals, these morphological differences diminished, suggesting that sexual dimorphism is less pronounced, potentially due to the biomechanical forces of bipedal locomotion. In the MUSH sample, reliable sex estimation was not possible, as most individuals fell outside the morphospace of male and female reference groups, hindering the exploration of sex-related biological and cultural variations among MUSH infants. So, this study is the first to apply 3D geometric morphometrics to the auricular surface for sex estimation in non-adults. It uses a non-invasive method with virtual replicas, aiding in the preservation of osteological collections. By using 3D models, this technique can capture and quantify variations in the auricular surface that conventional morphometrics cannot. This method can be applied to human skeletal remains from various contexts and periods, whether they are articulated or not. However, this study has limitations, such as underrepresented age categories in the reference sample, which were addressed by grouping individuals into 3-year categories. The method's effectiveness in other osteological samples remains untested. While promising for estimating the sex of individuals under 1 year, caution is warranted as sexual dimorphism patterns may vary between prehistoric and contemporary samples. The analysis focused solely on the auricular surface for non-adult sex estimation. So, given the importance of human osteological remains in understanding past populations, these findings require further investigation. Future research should analyze more iliac features and incorporate a larger sample to examine intra- and interpopulation differences. Moreover, cross-validation and cross-application with other archaeological samples and the use of AI techniques should be considered. So thank you very much for your attention. And I would also like to thank the institutions and projects that funded this research and the organizing committee for accepting this presentation. Thank you very much.